Hello, everyone, and welcome to today's webinar, Changes to USP 1788, Subvisible Particle Matter. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit them at beckman.com. Now, we encourage you to participate today by submitting your questions you have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, if you have any trouble seeing or hearing the presentation. I'd now like to welcome our speaker, Tony Harrison, Senior Marketing Manager at Beckman Coulter Life Sciences. Tony, welcome. You may now begin your presentation.
Thank you very much for that introduction, and welcome everybody to this short presentation on the revised United States Pharmacopoeia Chapter 1788. It's called the Determination of Subvisible Particle Matter. As our company name, Beckman Coulter, suggests, we did invent the Coulter counter in the first place, and we're also the inventor of the HIAC brand of light obscuration counters for subvisible particle analysis. And both Coulter and HIAC are named in the revised Chapter 1788 and 1787 as the preferred techniques for subvisible particle analysis. Now, the USP revised these chapters in response to the increasing number of biologic drugs in use and the need to differentiate between inherent particles that may be formed as part of the product characteristics and also those potentially harmful particles that may have been introduced to the product from sources such as packaging, processing, filling equipment, and of course personnel. The focus of this chapter is on subvisible particles in the range of 1 to 100 microns in size. The HIAC light obscuration test remains the preferred compendial pass-fail test method and backed up by the microscopic membrane method where required. This revised chapter introduces for the first time the orthogonal or complementary flow imaging test method as a useful complementary tool to these methods, and it emphasizes its use for characterizing the particle load in products and how it can help with forensic examinations to identify the contaminating particles to support product improvement, of course, as well as root cause investigations. So here's today's agenda. Today, we'll take an overview of the revised United States Pharmacopoeia Chapter 1788, and then we'll move on to take a look at the compendial pass-fail test for subvisible particles in injectable drugs, the HIAC light obscuration test. There are multiple USP chapters on subvisible particles in parenteral products, and we'll discuss the calibration and measurement ranges covered by this chapter. We'll take a look at some of the examples of particles that you may find in your products, and then we'll take a look at the complementary orthogonal Coulter counter and laser diffraction methods as called out in the revised complementary chapter USP 1787. So first, let's take an overview of the revised 1788 chapter. The revised 1788 became official on the 1st of May of this year, 2021, and 1788 is, of course, in the 1000 chapters, so it's an informational chapter complementing the chapters 787, 788, 789, and of course, as I mentioned already, 1787. Chapter 788 is harmonized with the European and Japanese Pharmacopoeia chapters on subvisible particle matter in injections, where the HIAC light obscuration method remains the official pass-fail technique for particulate matter. But for the first time, 1788 introduces the idea or concept of flow imaging as a complementary orthogonal test to the HIAC light obscuration method. And that's the compendial test fail method, but the flow imaging is a useful complementary tool. It provides orthogonal information to help characterize the particle load in products, and it can also be used to help with the forensic examination to identify contaminating particles, to support product improvement, as well as root cause investigations when you have a fail of the HIAC light obscuration test. 1788 encourages users to develop a strategy of particle determination in order to identify the category, if not the identity, of the particle population, so as to build a comprehensive control strategy for final production. So now let's take a look at that compendial pass-fail test mandated in the USP, the HIAC light obscuration technique. The HIAC light obscuration test method was introduced to the USP in 1985, and it remains the compendial pass-fail testing method. The HIAC method normalizes irregular shaped particles into equivalent diameter results, as if they were circular particles, and this eliminates the human error issue with microscopic membrane particle counting. Excuse me. A laser light shines through a measurement view volume and lands on a detector, which turns the light energy into an electrical signal. The sample under test is drawn through the measurement view volume. As particles in the sample pass through the measurement view volume, they block or obscure a portion of the laser light. And this reduces the light falling on the detector and causes the detector electrical output signal to drop proportionally. By comparing the size of the drop in the electrical signal to that caused by calibration beads, which are certified and traceable, the HIAC can now report an equivalent circular diameter for that particle, no matter what shape that particle is. This allows the HIAC to be calibrated with traceable certified beads of known sizes and to standardize this test method across the industry. As 1788 supports the chapters 787, 788, 789, it gives guidance on the recommended sizes of calibration beads used to calibrate the HIAC counter. Although the compendial pass-fail tests in 787 and 788 reference limits for the number of particles at just two size thresholds, greater or equal to 10 microns and greater or equal to 25 microns, this revised 1788 chapter suggests there is value in evaluating the particulate load in the sub-10 micron range and recommends that routine testing using the HIAC light obscuration method be extended to investigate particle loads down to 2 microns. And it's just adding measurements to greater than equal to 2 micron and greater than equal to 5 microns are also very useful. And it points out that the measurements at 2 microns are really a great indication of the quantity of protein aggregates in a biologic. As the chapter for ophthalmics, 789, also requires particle sizing and counting at greater or equal to 50 micron, it also suggests that data in the greater than 100 micron range is useful too. So, it goes on to suggest that calibration of the HIAC using traceable spherical beads at 2, 5, 10, 15, 25, and 15 microns in size. So, onto the recommended library of particle data. As can be seen by these images from a scanning electron microscope, particulates are very rarely uniform in shape. They're not spherical. 1788 recommends a strategy of a holistic use of a HIAC and orthogonal test methods right from drug development and continuing to final product QC in production in order to build a library of reference particle data to help technicians with particle classification, risk assessment, and even identification of these particle substances. Now, the revised 1788 chapter refers to the complementary chapter, which has also been revised, 1787. Both were made official on the 1st of May this year, 2021. Now, 787 recommends that the Coulter counter and laser diffraction are regularly used alongside the HIAC light obscuration test to gain more information on particle size and distribution. So let's take a look at the Coulter counter principle. Although flow imaging and microscope method are recommended as orthogonal test methods to the HIAC light obscuration method, the revised 1788 chapter is clear that reported particle sizes will vary between these methods due to the different measurement technologies used. In each case, the imperfect, well, certainly in light obscuration and laser diffraction, the imperfect method of using two-dimensional information to describe a three-dimensional object, it's bound to lead to variable results being reported, with further complications arising in samples containing heterogeneous mixtures of particles, i.e. a population of varying particle sizes. Now, in the Coulter counter, however, the particle's three-dimensional size is measured in terms of the volume of the particle. A voltage is applied across two electrodes immersed in a conducting solution, such as a buffer solution typically used for protein therapies. The electrodes are separated by a precision aperture. The sample in the test is drawn through the precision aperture between these electrodes, and particles present in the sample will partially block the aperture as they pass through, and they displace a volume of the conducting solution. In response, the Coulter increases the applied voltage between the electrodes to maintain a constant current through that aperture. And the size of the increase in the voltage is directly proportional to the volume of the particle that's partially blocking that aperture. The Coulter counter provides not only the equivalent spherical diameter, but also particle volume, and as such, the Coulter is very sensitive to small variations in particle radius because, of course, volume measurement represents the cube of the particle radius, i.e., volume is 4/3 pi r cubed. So any small changes in the radius are multiplied by a cube law, making them easier to detect as a volume change. Now, coming to our last topic today, we'll take a look at our laser diffraction, the third complementary method described in 1787 alongside HIAC and Coulter. Now, laser diffraction is an ensemble technique, so it measures a population of particles all at the same time instead of the other two, which is one at a time. So it gives a distribution of the particles present in the samples under test. Now, one weakness with laser diffraction used by itself is that smaller particles and a mixed heterogeneous population of particle sizes can remain undetected because the bigger particles create so much light diffraction. So to overcome this, the weaknesses, laser diffraction can be combined with polarized intensity differential scattering system, which provides particle sizing distribution information for those samples containing heterogeneous particle populations. And this allows for the detection of sub-1 micron particles in the presence of particles in the 10 to 100 micron range. So, in conclusion, the revised Chapter 1788 encourages users to develop a comprehensive strategy of particle determination in order to identify the category, as I said, if not the identity of the particle population, so as to build a comprehensive control strategy. The HIAC light obscuration test remains the compendial pass-fail test method, and Coulter, laser diffraction, and flow imaging are recommended as orthogonal complementary methods. So that brings me to the end of my very short presentation today. I know it's a very high-level overview, but I'd like now to open the floor for questions.
Thank you, Tony, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's take a look. It seems we already have some great questions coming in, Tony. Our first audience member says, "Thank you for your presentation. Please, can you explain the difference provided by the results between the HIAC method and the Coulter and laser diffraction methods?"
Thank you. No, thank you very much for that question. It's a good question. So, as we said, the HIAC remains the compendial pass-fail test. So this is the one that you're using your batch release testing on, and it's used for product release, as I said, in USP, EP, and JP. It does eliminate the human error in using a manual microscope method, which it superseded to size particles, but it only reports equivalent two-dimensional particle diameter. The use of this instrument is harmonized, as I mentioned, across USP, EP, and JP. So the equivalency between the reported pharmacopoeial test results has been established between these two test methods. However, the Coulter counter, on the other hand, measures the volume of the particles and calculates the equivalent 3D spherical diameter. And this makes it much more sensitive to small variations in the particle radius. However, its measurement range is limited depending on the aperture size chosen for the measurement. So multiple measurements are typically required when you have a mixed particle size population. It also reports particle counts, just like the HIAC, but the laser diffraction method, on the other hand, is a dynamic particle sizing technique that can measure and report particle size distribution, but not counts, in a sample containing particles from 10 nanometers up to 3,500 microns, just from one measurement. But as I say, it does not report particle count data, just distribution.
Thank you so much, Tony. Now, if the compendial pass-fail test particle sizes are 10 and 25 microns, why is the revised USP 1788 recommending the HIAC is calibrated from 2 microns up to 50 microns?
So what the USP 1788 is trying to encourage the development of a particle sizing and classification identification strategy, not just a pass-fail compendial test. So based on knowledge from the orthogonal testing of particle populations in formulations, users can build a picture of trends within the product populations that may indicate product is trending out of compliance before it fails the HIAC test. And this warrants further investigation. As I mentioned several times, the HIAC remains the compendial gatekeeper, if you like, for pass-fail testing. But if you calibrate and configure it to report particle concentrations from 2 microns up to 50 microns and above, it can give early warning of a potentially out-of-compliance trend in product quality in manufacturing. And this will prompt further orthogonal testing to get to root cause before the product goes out of compliance.
Thank you so much, Tony. We have one more question coming in from our audience. Can you please explain why the colored light scattering is required to enable laser diffraction method to accurately report a particle load of mixed sizes?
Of course. In order to measure the small particles, particularly in a population of both small and large particles, you need to capture the side scattering data from a polarized colored light source. And that's what the polarized system does. This data is fed into the calculation from the laser diffraction of the particle population, and it allows the instrument to know there are smaller particles there and to accurately report particles that are less than 1 micron in the size of a mixed population of much larger particles. Otherwise, you wouldn't see them.
Tony, thank you so much for your presentation today. Would you like to provide any final comments for our audience before we go?
Thank you. Yeah, I think this chapter is really interesting because it changes the view of HIAC testing from just final QC testing for the compendial pass-fail test, right through to pushing it upstream into the drug development and process development space. So the HIAC now should be used all the way through. And as I said in the presentation, it names Coulter as a good orthogonal test for more information about those particles, and introduces laser diffraction, which is good for the research and drug development, process development stage, but also it suggests that these Coulter and laser diffraction can be used in the GMP space, GMP manufacturing space, for particulate analysis as an orthogonal test to the HIAC in the QC testing.
Tony, I want to thank you again for your time today and for your important research. And I also want to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. And before we go, I want to thank the audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.